Hey guys, this is Mike Wolf. Welcome to the podcast. Today's guest is Pete Rowe, the CEO of Deep Branch Biotechnology, and their company makes technology that allows you to basically pull protein out of thin air. And you may be saying, hey, you talked about that recently on a podcast. And I did. But actually, the reason I had Pete on the podcast is I wanted to talk about CRISPR. I've been fascinated, like many, about this idea of using gene editing to create new and innovative foods, to help make more sustainable foods, to rethink how we do agriculture. And so when I talked to Pete at Seeds and Chips in Milan, and he told me that was his focus when he got his PhD, and it's some of the things that they're working on at Deep Branch, I said, let's come on the podcast. Let's talk about it. So this podcast is about that. We talk about CRISPR, we talk about gene editing and the implications for the future of food. And so if you're interested in that, if you wanted to learn about it, Pete does a great job of describing it. And I really appreciated his going into this very technical topic and explaining it to me, and hopefully you find it great as well. So, but before we get to our conversation with Pete, if you haven't already checked out the Smart Kitchen Summit in October, ticket prices are going up soon. So I would check it out. Go to smartkitchensummit.com. Use the discount code podcast. Also, if you are a food tech startup, if you are a future of food startup, we are actually having a startup pitch competition. 2 of them. One for food tech, if you're doing interesting food tech. Another if you're creating a future food product for consumers, and it's really innovative. And the prizes are great. We have Barilla, Blue 1877. We have WeWork Food Labs, both being sponsor partners in terms of like some of the prizes that you can get. But also, quite honestly, I wrote about this in a post about our startup showcase. If you win this thing, it propels you into the limelight. Like we just seen past winners have so much momentum after winning the startup showcase or even participating in the startup showcase. So I think it's a great opportunity. Not only do you get to participate, you get a free ticket to the event. You get to showcase your product. So go to smartkitchensummit.com. The front page, you can actually see the application. So check it out. For the rest of you who just want to come and see these startups, like I said, use the discount code podcast. We hope to see you there. All right, folks, let's get to the conversation. Hey, I'm really excited to have Pete Rowe of Deep Branch Biotechnologies on the podcast. Hey, how you doing, Pete?
Yeah, great. Thanks, Mike. How are you?
Great. We met in Milan, which is a great place to meet anyone probably. We were at the Seeds and Chips conference, and I passed by your booth, and I was intrigued because I had heard of you guys, because I'd gone and done a deep dive fairly recently looking at gas fermentation, this idea of creating protein out of thin air. And this is something that you do. And we're going to talk about that. We're going to also talk about CRISPR and gene editing as well. But tell us quickly what Deep Branch Biotech does.
Yeah, that's right. So like you said, we met in Milan at Seeds and Chips. So it's a big ag tech conference. So that kind of gives away exactly what it is we do in terms of which fields we interact with. So as a company, we take carbon dioxide directly from industrial emissions and convert it into single-cell protein. And this protein we use as a partial or complete replacement for the conventional proteins that you would find in animal feed or aquaculture feed. So things like soy or fish meal.
Which I love this idea, right? So this idea of not only are you're taking kind of bad outputs that would normally go out, industrial outputs, into the air, and we're trying to figure out what to do with that, but you're actually using it as an input, and you're also doubling down on the good effects, and that you're not creating a tax on the earth in the sense you're using these inputs rather than some other input.
Yeah, that's right. I mean, so I guess circular economy is quite a lot in vogue at the moment, if you will. And really, that's a great way of describing what we do as well. Without it being a cliche, we take what would be considered as a waste stream conventionally, and we can reimagine that as a valuable resource. And the way we get value out of it is indeed by turning it into protein.
And we were talking about what you guys do, and you mentioned that a lot of your academic research was on gene editing and CRISPR, and you're starting to think about using some of those thoughts and research around what you do at Deep Branch. But tell us a little bit about your background, what you studied, and what you dive deep into.
Yeah, so that's right. I mean, in terms of my role right now, I'm the CEO of Deep Branch, so I have a commercial role largely. Of course, it's a startup and people wear many different hats. But prior to this, I actually have a technical background in genome editing. So for 4 years, I spent a lot of time working on CRISPR-based technologies, and specifically applying these genome editing technologies and developing new ways of applying them into gas fermenting microbes. So at Deep Branch, we use these microbes to generate protein from CO2, like we just described. But during my sort of academic career, I was looking into new ways that you could utilize these for a similar process, whereby the output was fine chemicals and platform chemicals rather than protein. So essentially what you use genome editing for in this context is what you would regard as metabolic engineering. So changing fundamentally the way the cell's metabolism works so that you can entice these organisms to create high value compounds. You then ferment them on your gas stream and then extract these high value compounds and use them usually as alternatives to traditionally petrochemical derived compounds.
I want to step back a little bit for our audience, because one of the things I've just been fascinated with is this idea of gene editing of CRISPR, but it's a fairly technical thing to approach, right? And something that you spend your years studying from an academic side, you're looking to commercialize it now. But I think for kind of a general audience, even in food tech, CRISPR is fairly new. So can you give us the 101 on what CRISPR is and how you describe it to people?
Sure. So, I think it's important to take maybe even another step back before going into what CRISPR is and perhaps give people a reintroduction to maybe genetics 101. Yep. So, a gene is a unit of heredity. In a molecular sense, it's a sequence of DNA, a genetic code on your genome. And generally speaking, what genes do is they code for proteins which have some kind of cellular function. So different genes have got different functions, and particular genes maybe have different varieties, which are called alleles. So like I say, these genes code for proteins with specific functions, but these alleles, so these different flavors of gene, code for proteins that do pretty much the same thing, but maybe in a slightly different way. So there's a natural level of variation within your genes. The most obvious example that most people use is that of eye color. So there's a big genetic component to which color your eyes are. And the reason why I have blue eyes and my wife has brown eyes is because this is a slightly different allele. And the likelihood of our kids having blue eyes is relatively slim, because brown is the dominant one there. So we'll come back to why that's important later on. But really from a food angle, it's clear that genes are important because they code for proteins that can impact things like taste, and color and texture and resistance to genes. So a whole plethora of different aspects of food production, from the production of the crop and performance and yield to taste and more sort of consumer relevant concerns. And I mean, it might all sound quite abstract, but really what genes do is in a large part determine the characteristics of what it is that the plant, in the instance of plants, or the animal, if you're talking about livestock production, the genes that these organisms have will determine how they behave and ultimately, in the context of food, what they taste like. And this is where CRISPR comes in. So it's a really powerful way of modifying or selecting for particular genes or particular variations of those genes. So CRISPR is actually, or CRISPR-Cas systems are the defense systems employed by bacteria. So just like us, bacteria are prone to infection, and they are prone to infection from things called bacteriophage, which are a kind of virus. And although we have a relatively complex immune system consisting of, lots of different factors, the bacterial immune system is really simple. It's basically just a way of cutting viral DNA, because viruses are just essentially DNA wrapped in a protein coat. And when they enter the cell, they become just naked DNA. So microbes, bacteria, evolved a way of cutting this DNA. And so within the last decade or so, people have realized that this is the case. And some very clever people realized that rather than having these systems geared towards cutting viral DNA, you can repurpose them to cut whichever kind of DNA you want. So then it becomes a really powerful tool, because you can use it in the context of plants. You can cut DNA which would encode for a particular kind of gene, and use the technology to either remove that gene completely, or swap it for a different variant, select for a different variant, or even insert new DNA into the cell by using it as a selection measure. So it's basically an extremely powerful tool of making genetic manipulations to cells, cells in which would form an organism. So you can think about that from a very simplistic point of view. So you can use them in the kind of context that I develop tools for, and what we do at Deep Branch Biotechnology, which is using them for single-celled organisms. But they're equally powerful, or even more powerful, in using them with multicellular complex organisms like plants or animals or even humans.
And I'm glad that you brought that up, because there's just been a broader conversation as the certainly the scientific press, and then kind of now it's distilling down to the more mainstream press, what this means. Part of the conversation becomes somewhat hyperbole. There's all sorts of kind of excited headlines about this idea of like gene edited people, for example, and certainly there's a whole scope of different applications that you can do. You're primarily, and we're primarily talking today about food applications. And even within that context, there's a whole bunch of things you could start to talk about, right?
Yeah, I mean, it's huge. Within plants alone, I think, if we just put animals and humans to one side for now, it's massive. So it's everything from, getting a nicer color to your tomatoes, to improving their performance in terms of disease resistance or shelf life. Some of these components have a relatively simple genetic mechanism behind them. So you can know by changing one gene that you can actually improve the shelf life significantly without it impacting on taste. And this is really important because genome editing can solve some problems that conventional plant breeding can't. So often what you find with plant breeding is that if you're selecting for a particular trait, it will drag through other traits with it. And this is for reasons that I won't get into. But essentially, it's often quite hard to divorce impacting on one aspect without it dragging through an impact on another. Whereas with a very specific tool like CRISPR, you can divorce those two things. So for an example, this is completely hypothetical, but let's say the best tasting avocados also happen to be the ones that bruise easiest. So you had extremely tasty avocados that you couldn't really ship around the world, and therefore they weren't really suited to large-scale agriculture. But if you can make a specific genetic change so that the flavor benefits you get, but you also retain the conventional longevity of this avocado on the shelf, then it's kind of the best of both worlds. And there are some very complex, clever things that you can do with CRISPR. But I think a lot of applications that are important that people are talking about are relatively simple, as simple as just speeding up the breeding process. So that there may be a particular genetic component that's been known for decades. And, people crossbreed plants all the time to get specific traits that they're looking for, but you can massively speed up that process. So beyond taste and flavor, there's also things like disease resistance. So if you think about all these problems that people are having with coffee, so coffee rust is a kind of fungus that infects coffee plants. There's also a big genetic component in that. If you make one particular genetic change within the coffee plant, the likelihood is, if you know what that change has to be, that you can stop that fungus being effective at infecting the coffee. And, that's really important in terms of flavor as well, because, I quite like coffee myself. So if you look at the difference between Arabica and Robusta there, it's another trade-off. You have the much better flavor profile that you get with the former, but with the latter, you get much better disease resistance and hardiness. And conventionally, the two can't be seen as going hand in hand. But the power that CRISPR has is you can ultimately chop and change and have something that could, in theory, be extremely hardy, but also really, really flavorsome. And then you could get into some more complex aspects of it as well, whereby you can use the technology in the kind of way that I was doing for developing different kinds of chemicals. You can do metabolic engineering. So you can go within the coffee bean and say, okay, we recognize that these particular aromatic compounds are really good in terms of flavor. So let's overproduce those and have a super punchy, really nice floral, citrusy coffee that you can have as a nice sort of in your AeroPress, if you will. So, there are a whole different spectrum of ways in which you can use CRISPR that vary in terms of complexity. And I think the problem with regards to the regulatory aspect of that, particularly in the EU, where last year there was a landmark ruling on this, is that the current regulations on genetic engineering in plants don't reflect that space for nuance that there is in the application of these technologies.
So with regards to that, real quickly, you're kind of highlighting the difference between the regulatory environment in the EU versus US. USDA is taking a much more hands-off approach with regards to gene editing relative to the EU, and they see it as different than GMO, which I think. So I before we jump into that, can we step back and kind of like talk about GMO, genetically modified versus gene edited? Because I think that's an important distinction that people should understand.
Yeah. So I think a lot of the applications that people are talking about and actually using CRISPR for today are they're using it for a purpose for which other tools have been available for the last couple of decades. But the difference there is kind of twofold. One, it's much more efficient to use this new generation of CRISPR-based tools. But secondly, if you use them in certain applications, it's scarless. So whereas if you had something like a GMO crop in the 90s, you would have to introduce foreign DNA permanently into the genome of that crop often in order to have the kind of change that you want to make. So you couldn't escape the fact that you were modifying an organism, putting foreign DNA into it, and then it's out there. It's in the wild, and there are sort of different ways in which you could try and contain it, but ultimately there's a high risk there. And the reason why the US with the FDA are taking a more hands-off approach with regards to use of CRISPR in some ways is that as a technology, what it can do is delete specific genes in a way that doesn't leave behind any foreign DNA, so that it's not bringing anything into the wild that isn't already there. It's simply removing a piece of DNA that was there. So the likelihood of that having a huge impact, uncontrollable impact, is relatively slim.
What about, real quickly, and this is for a layman, it's deleting certain components or certain aspects of a gene. Is there a cut and paste aspect, right? So you had the idea of like Arabica beans and another variant of coffee where they each uniquely had their own beneficial characteristics, taste versus robustness in terms or whatever, or disease resistance. If you're combining those within a certain gene sequence, A, can you do that? Again, this is a layman asking. And B, is that different than like inserting a foreign DNA, a foreign element?
Yeah. So what you just described there would be, recombinant DNA technology. Okay. Maybe for that instance, there's
and if it isn't evident to my listeners already, I'm not a scientist. This is more just
yeah. So maybe that one might be a more gray area in terms of an example because it's two varieties of coffee. But let's say to make this a bit clearer, if we were using some flavor aspects of the cocoa plant that you used to make chocolate with, if you're using some genetic component of that and putting it into a coffee plant to improve your flavor profile, this would be exactly what we're talking about. It would be foreign DNA that wouldn't naturally be occurring, being put in a recombinant fashion into a plant, and that plant gets planted into a plantation, and then it's
real quickly. That would be more widely kind of viewed upon by the scientific community as genetic modification versus something that's more gene editing.
Yeah, precisely. So if you think about it, that's kind of a bit more playing God than just simply deleting a gene, because it can have wider implications of if that plant breeds with another form of in this instance a coffee plant, and then it has a different progeny which has different characteristics, that then it can really spiral out of control. But even that is a lot safer than the previous generation of technologies that were available. And the reason for that is often, if you think about the less powerful forms of genetic engineering technologies that were around before CRISPR, often these were reliant upon using a selective marker. So rather than having this really powerful set of molecular scissors that cuts DNA, which is what CRISPR is, and anything that gets cut gets killed, when you're talking about using other forms of older kind of generations of genetic engineering, you were often reliant on inserting, let's say, a pesticide resistance gene into a specific gene that you're looking to delete. By kind of ramming it in there, it disrupts that gene, and the protein that a gene encodes for no longer works. But of course, what you've done there is you've bunged in a pesticide resistance gene within the genome of that particular sort of commercial crop that you're growing. So then, once that's out there, you've got a crop that's resistant to pesticides. And if there's chance of that gene spreading in the environment, then there's a real risk. So this is why people are saying CRISPR is a completely different thing, because you haven't got that reliance on a foreign gene as a selection measure. All the changes that you're making to the genome are purely functional with regards to what you want the plant to do, rather than being part of the genetic engineering system that you're using to change them.
As with a lot of things, right? There's an evolutionary path where like early tools were much more brute force, kind of a hammer to the nail kind of analogy. And what we're seeing now is much more high precision tools, less harmful. But what I'm wondering is, so getting back to the European Union conversation, they do take a different view on gene editing, and they and maybe correct me if I'm wrong, they do still seem to bucket it under the same category as GMO relative to the USDA, correct?
Yeah, so I think if you look into the reasons why they made that decision, it actually makes sense, because what they're saying is it would be a case of opening the floodgates. So if they said, okay, all CRISPR-based technologies are okay to use, then that would also include technologies that are making huge changes to the genome and inserting foreign DNA and doing things that could potentially be really scary. So I think ultimately what we will see within the EU would be a changing regulation that would reflect the different spectrum of ways in which you can use CRISPR-based technology. So you got the more safe, simple ways of using it, like I said, to speed up the breeding process, or deleting specific genes associated with,
negative characteristics.
Yeah, particular negative characteristics like not being resistant to particular diseases. You know, if you're using it for things like that, then it's clear that it's really not going to have a huge impact in theory. Whereas if you're making big changes to the genome, then it's a lot less clear. But how you can have regulation that reflects that, maybe it becomes a bit tricky. You know, does it go on a case-by-case basis? Is that itself going to slow down ways in which we can go about doing this? And then, I mean, the obvious problem there you have within the EU is you don't want to have a situation where Europe gets left behind. If you have a much more proactive stance on genome editing and you're already using it for a lot of good in different parts of the world, then you've got the EU, which has conventionally been a leader in terms of agriculture. If you think about the amazing work that the Netherlands did in the post-war period and still continue to do, you're essentially, boxing yourself out of a really large aspect of the future of agriculture. And I think, if you think about that on an even bigger level, the fact that we need to feed 10 billion people by 2050, if it is a case of using these technologies to just improve yields or improve resistance to diseases so that for each square meter of land, you're getting a hell of a lot more output of crops, and of those crops, a lot more of them are going to the fridge of you or I. That's ultimately going to be a really good thing and something that's extremely necessary.
Real quick, can I ask a question on that? As we look towards global warming, right? If we just accept the reality that the climate's changing, that certain, we talk about the danger of coffee dying, or like maybe will we ever have bananas 10 years from now, right? climate change is real with regards to some of these specific crops, could you do gene editing to make certain breeds of plants that are in danger resistant to the change in the climate?
Well, I think,
or adapt to the change in the climate, right?
Yeah, I think one of the best examples of this I've seen would be having salt tolerant rice. So, in a conventional rice paddy, you'd use quite a lot of fresh water in order to grow the rice, which really limits your capability to grow rice to places where you have a lot of fresh water. And there are some studies that have been done showing that you can do particular targeted genome editing on rice to create new variants that are capable of growing in brackish water, so water with like a relatively high amount of salt, or even I think they're pushing for sort of seawater. So if you could have that, whereby you can grow rice almost anywhere, that would be fantastic. And in terms of going forward, if you take global warming out of the picture for that, then it's of course answering the question of how do you produce more in areas that wouldn't traditionally have been suitable for arable land. In terms of climate change, I'm sure there's a particular genetic component to a lot of plants' ability to grow in particular heat. But I'm not an expert in terms of plant biology, so it may well be a little bit more complex.
So you're saying we have a chance.
Yeah.
To quote Jim Carrey.
and this again is also the power of CRISPR in the fact that it's a lot quicker at being able to answer those questions.
Yeah.
So if you do think there's a particular genetic component and a particular target within the genome that you think can be responsible for drought tolerance or resistant to higher heat than what the plant would conventionally be able to thrive in, you can use the technology to really quickly answer that question. So I think there's a hell of a lot of low-hanging fruit probably out there that you can use CRISPR to really realize. And it may well be that some of the things that we think would be easy to solve wouldn't be. But similarly, it may well be that there are lots of unknowns out there that it turns out it's really simple to make a really impactful change.
The market is already seeing gene-edited products injected into the food system, right? So there's a company called Calyx that actually has created a soybean that basically edits out trans fats. And that's really kind of the first one that we've seen, at least in the U.S. market, that is going to be available in restaurants. We've seen lab-grown cellular meat guys like New Age Meats, they're experimenting with it. So we're certainly already seeing this happen, right? We're already seeing folks experiment with this type of technology for products that people will be eating fairly soon.
Yeah, and I think that's really exciting. Like I say, the era of CRISPR is upon us, and it's just a question of how we're responsible in using this technology. I think, as a technology, it's come about as a culmination of lots of different aspects of cellular biology, molecular biology coming together and having this really nice tool. But with that, you've got other powerful techniques like the ability to sequence whole genomes relatively quickly. So whereas before in the 90s, you would have been worried about what big changes to the genome could have been made that you don't know about from using particular genetic engineering techniques, you have this extra level of safety available now from other technologies. So it'd be really quick to, in the instance of the soy that you've described there, you can literally look at every single base pair within the genome of the plant that you've made and just quickly check that there's no other changes that have been made that you didn't want to make. Because, that's one of the big fears of using CRISPR-based technology in humans particularly, and in a lot of complex organisms, is that sometimes you have what are called off-target effects. So you tell the CRISPR system to go and cut a particular region of DNA, and it will go and do that. But what you might not know is that it's also cut somewhere else on a different chromosome, so on a different region of the genome, which will have a completely different effect. And, we were talking about answering those complex questions and doing an experiment. It could well be that the gene that you've made a change to, and you can test for that and say, yeah, that's definitely been deleted. But the changing characteristic that you've seen in the plant might well be because you've made another change somewhere else in the genome that you don't know about. So I think it's really about finding the synergistic technologies and making sure you can get them to work together. So, with that level of safety is available if you use the right complementary tools.
Let's talk a little bit about how democratized this technology becomes. It sounds like something that only scientists in a lab are really going to be able to do in the near term. But what I think could be interesting is downstream. Can there be tools that can essentially democratize this for people who are basically working at CPG companies, right? And then ultimately, maybe 15 years down the road, can you allow makers to kind of do this type of thing who are maybe not working in labs as much? Does that make sense? Like, can you ultimately create tools as this becomes more evolved to allow people to create gene edited food or customized around specific tastes, or to put products out to market faster?
Well, I think this is probably a position that I would take a more kind of European stance on, in that it's an extremely powerful technology, but in the wrong hands, it could be quite dangerous. That being said, this stuff is already out there in the public domain. It's a lot of the power of technologies like CRISPR are that they are really simple to use. Obviously, from a layman's perspective, it will take you a while to get your head around it. But the ease in which you could have in making changes to a genome today, just infinitesimally, they're just hugely, it's way more simple to do it today than it would have been 20 years ago. So the possibility of people doing it is there. Whether or not you have sort of the ability for governments to intervene in people doing that is another question. So, you're seeing things like, I don't know if you're familiar with it, but there's a British company that produce basically a USB stick that can sequence a genome. They're called Oxford Nanopore. And with that, it's an important part of having a at-home lab that you can get for I think it's $1,000 or so. So the ability to do this in your own maker lab is probably quite simple. But
so it's already been, to a certain extent, democratized.
To a certain extent, yes. But then the question is, what is going to be the impact of doing that? Right. It's the kind of thing where the reagents to do this, I could probably source them all myself personally and do it at home because I know what I'm doing. But the danger there, I'd say, is actually, maybe I'm biased because I'm a microbiologist by training, but if you think about the ways in which you could genetically engineer particular strains of bacteria to be a lot more pathogenic, that's where I really see the risk being.
Right. You put these in the wrong hands, people with kind of ill intent or malice, they could create powerful things that could be put into the world that you don't necessarily want put into the world. Yeah,
exactly. And like I was saying about the older generation of the old generation of genome or genetic engineering tools, once the genie's out of the bottle, you can't put it back in. If you've made these changes and you've got a very dangerous organism out there, it's very hard to try and stop it spreading. So, I think to a certain degree, you've got to be very sensible about the way you do this. But then the other aspect is there's always going to be bad actors in the world. People with malintent that want to use these technologies for, not the best intentions are always going to be there. And it's a question of how you can stay one step ahead and ensure that everyone is playing by the rules. And that's a question that I don't really have the answer to, I think. And maybe a lot of people would struggle to have that.
That gets into like almost philosophical questions, and we could certainly have those more esoteric conversations. But for today, I think we can kind of sideline that. I do want to ask this. What is the potential for CRISPR in CPG products like, packaged food? Like, can I get like a super healthy Hot Pocket or Captain Crunch at some point? Like, can you like create like ultimate food that tastes exactly like you want it? You think it has all the bad things in it and it's horrible for you, but actually is healthy? Or is that just kind of like a dream of mine that will never happen?
Well, maybe I'm not the best person to answer this, but from my understanding, it would be that the things that make those kind of foods tasty, the saturated fat, the sugars, the carbohydrate content of them. That's something that genome editing can't really answer.
Okay.
But I may be right. A good example there would be if you look at something like stevia. There's quite a few companies that are looking into ways in which you can produce that via fermentation rather than by a plant, which would mean that you can get, in theory, in a few years' time, you may well be able to produce something like stevia on a really high scale. So you can have artificial sweeteners that are price competitive with sugar, and therefore you can push it into a lot more products. And maybe that's a good thing, but maybe there are negative health benefits with having something that you've not that we haven't evolved to eat in high doses. So I think it's an interesting one. But one thing that would be really interesting in that regard is if you were to change your microbiome. So one really powerful way that you can use CRISPR-based technologies is by changing the makeup of your gut, which obviously has a huge impact on how well you digest food. So again, it's a very complex field of science because there's just a huge amount of variables. You've got sort of thousands of different kinds of microbe in your gut. And
that science is fairly new too, right? Like we're just trying to. The idea of the microbiome is like scientists are just trying to get their mind around what that is at this point.
Yeah, exactly. It's a problem that people haven't even defined. So, answering that question, if you don't really have a well-formulated question in the first place, is always going to be difficult. But that being said, there are some, like I said before, low-hanging fruit that you can really speak to. So, there are particular pathogenic bacteria that we know in any quantity are bad to have in the gut and often quite hard to remove. So something like C. diff, which is a hospital-acquired infection. People are working on ways in which you can use CRISPR to just permanently remove that from the gut. If you think about ways in which you can deliver therapeutics that aren't going to have the huge impact of using antibiotics, that could be a really interesting thing. Things like, the whole point of CRISPR is that it's evolved from the ability of bacteria to protect themselves from viruses. But these particular viruses, people are also using them in the context of food protection. So if you think about something like raw chicken or salad, conventional means of getting, food poisoning, people are using these viruses that naturally infect those dangerous microbes, and they're spraying the food with these viruses, which just when they come into contact with those particularly harmful bacteria, they kill them. But the effectiveness of the virus to make that to have the ability to kill the microbes is something that you can optimize with CRISPR as well. So I think basically anything that's got DNA in it, you can use CRISPR for good. It's just a question of figuring out how you can go about doing that. And like I said, for some things that's really simple, but for other things it's extremely complicated. But the whole point of the technology is that you can really learn fast. You can have short iterations of applying the technology, figuring out what's up, and then going for it again.
Well, this is really exciting. Pete Rowe with Deep Branch. Tell us a little bit and quickly where people can find out more about your company.
Yeah, so you can follow us on Instagram or Twitter. So that's at Deep Branch Bio. Our website is www.deepbranchbio.com.
And thank you. And I feel like I should call you Professor Pete because you gave us this great lecture and a lesson and fairly interactive about what CRISPR is. I really appreciate you taking time and talking with me today.
Yeah, it's no problem. Science is actually, obviously I work in a biotech company, but it's something that I don't spend a lot of time talking about anymore because I have a more commercial role. But it's been very interesting to try and sort of spread some of my knowledge, limited as it is within the context of food. I think hopefully there's a brief taster into exactly the power and the breadth of what CRISPR technology can do there.
Great. Thanks, man.
Yeah, no worries. Thanks for having me.